In order to build a bioluminescent system, we're going to be using the pure cell-free assembly kit. The system will make use of the DNA sequence for the required proteins. Transcription factors will trigger the expression of the mRNA to start the sequencing proteins luciferase and furimycin. The system for bioluminescence uses the two proteins furimycin and luciferase. When bound together in an enzyme-substrate complex, they release a high-energy photon, causing the visible light you and I can see. Taken together, many reactions cause a more intense light. Its brightness is unlimited, and the system is environmentally friendly and recyclable with the addition of the ATP to the system. For our system, we'll be adding the complex into cyanobacterial encapsulins. These protein-based capsules allow us to keep separate the required components for the bioluminescent reaction. When subjected to pressure, the capsules break open, allowing the constituent parts to mix and begin the reaction.
To investigate bioluminescence, we have chosen the nanoglow luciferase system. First, E. coli cells were incubated overnight. Cell density was confirmed using spectrophotometry, and luciferase expression was initiated by adding a buffer solution. Because luciferase is intracellularly expressed, cells must be purified and lysed. The sample is first centrifuged. This spins the sample at a very high RPM to settle the cells onto the bottom of the tube, forming a pellet. The pellet is then resuspended in integral lysis buffer using vortex. As the substrate is added, the bioluminescence reaction occurs immediately. When the substrate levels are low, bioluminescence is hard to observe with the naked eye. However, under UV light, the intensity is obvious.
So we decided to design an emergency balloon which uses cell-free protein synthesis and nanocapsules. In the balloon, luciferase plays a substrate and luciferins are the enzyme, and when they are met, it will come into bright light. After we do the experiment in the lab, the emergency balloon seems not suitable because the lifespan of the pure system is around 24 hours and it is not enough long time for the net rescue. So we changed the direction of our product design, discover more design opportunity with the visible light because this is the direct connection with our bioluminescence system. Then we came up with several ideas. The first idea is the Christmas cracker. The reason why we want to design this because it plays an important role in celebration event and party. Also, the cracker needs to activate the pressure. And the bioluminescence Christmas cracker can provide the light in the night as for the decoration of the event, even for the birthday party, which is an environment-friendly product. The second idea is self-illuminated lamp, which can provide the bright light without any support. The self-illuminated lamp can provide enough bright and 24-hour stable light for people. Also can easily change the capsules, then can make sure to recycle the lamp, which is environmental protection. Also, the self-illuminated lamp is easy to bring with, and we designed this lamp as a folding lamp because this can break the capsule and activate the pure system. The advantage of the lamp is it have a good apply on the outdoor exercise in the night, and also can play roles as an emergency light when met no power resource environment, even no solar power. With the tutor and the group's help and advice, we try our best to find more application for the bioluminescence system. Finally, we figure out the innovation is to change the limitation into the advantage in our project. So we're thinking about the bioluminescence ink. This ink can light in 24 hours, which can have a mark on different object surface. And this will be a unique product for the child because they can draw anything they want in their home and their parents don't need to worry about the cleaning work. The pen is designed as transparent, and there is a small ball in the replacement part to keep the ink would not leak. The tip of the pen will be attached to a small cylinder through the sponge column. Upon press the replacement part to connect to the cylinder, the capsule will break by high pressure and the luminescence process will begin. The luminescence ink will vanish after 24 hours, and also we want to design the pen as a press tab which can provide pressure to activate the process and break the capsules. Then luciferase and fumarins will produce the intense light in a short time. The reason why we decided to design the bioluminescence ink is they have a huge market as for material and they also have lots of applications in the future because the bioluminescence ink can glow in a period and then fade away. All in all, our project has focused on the idea of using bioluminescence in a number of different settings. By designing cutting-edge biologically advanced cell-free system, we are able to make use of pressure-activated bioluminescence. We make use of pure cell-free assembly kits to manufacture the constituent part of the luminescent system. The system have a variety of different applications and using in our day-to-day lives.